Hi, my name is Soper, and I am I'm an assistant professor at the University of Dallas, and I'm going to be talking to you today about growth patterns and genetic expression of the HIPPO growth pathway in this endangered coral species. So stony coral has been under extreme threat over the last 30 to 50 years and has resulted in extreme ecosystem degradation. So we see here that in 1975, Carey's Fort Reef was beautiful. It had lots of aquapora palmata. There are fish in this picture. Sergeant majors are kind of hiding out underneath of some of the branches. And Carey's Fort Reef, just to back up a second, is actually a reef off the coast of Key Largo in the Florida Keys Reef Tract. And in the same spot in 2004, that's what the ecosystem looked like. So we have seen extreme degradation in the US, but it's also global. So on the left-hand side, you'll see a picture of the percent degradation of the Great Barrier Reef Tract. So this is a portion of the Great Barrier Reef Tract, and in the center you'll see red. That's where they've seen the greatest amount of loss. On the right, you see a graph where on the x-axis is the year, and on the y-axis is percent coral cover. And in 1985, they're at approximately 28% coral cover. And by 2012, they are down to around 12%. So this reef loss that we've seen is actually global. And it's due to not only climate change, as we've been talking about, Luke's talk earlier highlighted that fact, and of course increasing CO2 emissions, which has decreased pH, but also things like disease has been on the increase. About a year and a half ago, there was a new and emerging disease off the coast of Miami, and it was new because it actually has infected over 22 species of stony corals, and it's been quite widespread. So it just recently, earlier this year, reached the Florida Key West in the Florida Keys. So it's moved all the way down the Keys Reef Tract. This is a picture that I took on Lou Key, which is in the southern part of the reef tract in March of this year, and you see a brain coral in the center part. The green areas are the still living tissue, but most of that coral head has degraded and died. One thing we do see though, and as an evolutionary biologist, I was kind of had some level of hope, is that we do actually see a heterogeneity in response to stressors. So this picture I took on the Spiegel Grove in December of 2018 on the deck and of the wreck. And on the left-hand side, you will see that there is a brown little patch. That's actually a nice, healthy little piece of coral. And the reason why it's brown is because of there is an algal endosymbiont, sometimes called zooxanthellae, but they belong to the Symbiodinaceae family, and that is indicative of a healthy coral. The one in the center, though, you do see bleaching. So when the coral becomes stressed, they release their Symbiodinaceae algal symbionts, and they bleach. And if they bleach for long enough and conditions don't return to normal, then they are going to die because they cannot live without those endosymbionts. Restoration efforts have been continuing around the world, and there's a variety. I don't even have time right now to talk about all the different restoration efforts that are taking place. But to just name a few, on the left, there is a spider-like structure. This is in Indonesia, where they've been planting, and in the Coral Triangle in particular, they're using this particular method, where they've been outplanting patches of coral onto these structures, and they see an increased growth rate. On the right, this picture is something that they're trying to do to mitigate that disease I was telling you about earlier. You'll see those brown kind of like edges around the coral itself, and those is like a putty that is laced with antibiotics. We have seen a response to antibiotic treatment for that particular disease. But most recently, also, they've been doing something called microfragmenting. So this is Dr. David Bond. He is formerly the head of the field station for Mote Marine Laboratory on Summerlin Key. And a few years ago, he discovered that if you fragment coral down to just a few polyps, you microfragment it, it undergoes rapid growth. And so on the left here, you'll see that these are tiny little microfragments that had just been cut like the day before when I was there. we started talking about this data that he had collected with some colleagues that was recently published. On the left-hand side, you'll see the microfragments have been outplanted, and on the right-hand side, there's a larger fragment. And what they were doing in the study is they were looking at the coral cover and growth when they outplant back onto the reef tract microfragments versus larger fragments. And so on the left, you'll see that there is a significant change in and an increase in microfragments for the change in surface area. So he and I started talking, and I said, Dave, what are your biggest unanswered questions? And he said, well, number one, we don't know what genes are expressed and why and how this is happening. We know it happens, but we don't know how it happens. And so he sent me some samples of Orbicella faveolata. This is actually Orbicella in nature. On the left, I took that picture again on Luke in March. And then on the right is a polyp, one like zoomed in polyp of one of the samples that he sent me underneath of my dissecting scope. But before we can start answering questions about like what is happening or how genetically this is happening, we had to actually understand what tissues are undergoing rapid growth. Because our aim was to look at tissues that are undergoing rapid growth versus tissues that maybe aren't undergoing rapid growth on the microfragment itself. So being a microfragment is pretty small and sometimes difficult to work with, but what we did was we examined new polyp formation, and we did this by taking photos 3 times per week. We examined 1 fragment, 3 genotypes for both number of new polyps as well as polyp size. And so what we found was that on the x-axis, I was looking at 3 genotypes for each of 4 months post-fragmentation, 8 months post-fragmentation, and 16 months post-fragmentation. And we found that the number of polyps on the edge was significantly more than in the center. We basically don't see polyp formation in the center of the microfragment. All of the new polyps that we were seeing occur on the edge of that microfragment. We also looked at polyp size and saw that on the right, and in red is the average polyp size on the edge versus in the center. So there was a significant increase in, well, the center polyps were actually significantly larger than the ones on the edge. And this was true across all 8 weeks that we saw pretty much. There was a couple of weeks where due to varying, it's hard because we're working in a closed, Aquarius system. Sometimes, parameters go up and down and polyps respond to that. But overall, there was bigger polyps in the center than on the edge. So to answer the first question, new polyps form on the edge, as well as the edge polyps are smaller than the center polyps, indicating that they're newer. So then we went on to say, okay, how or what growth pathways are involved and how are they expressed? So we wanted to use a targeted gene approach, and we targeted the HIPPO growth pathway. And my colleague, who is actually a fly geneticist at the University of Dallas, said we need to use the HIPPO growth pathway because it is conserved across many different taxa, including at least one other cnidarian had been discovered in. It had never been found in corals, but it had been discovered in a sea anemone. So we knew that the HIPPO growth pathway was predicted to be in Orbicella faveolata, and so we decided to kind of target this particular growth pathway. And so the HIPPO growth pathway, when it's off, it actually initiates growth. You can see on the left-hand side here, there is a gap between these 2 cells. So when cells have intracellular space, this growth pathway is initiated. There's a whole bunch of stuff going on here, but ultimately what happens is it targets different transcriptional factors, like, for example, cyclin E. So cyclin E is a transcriptional factor that stimulates the S phase of mitosis. And so what we did was we developed 3 primers for Orbicella faveolata to target the cyclin E transcriptional factor. So what we did was we I dissected out this polyp here, this particular fragment, and then this polyp here on the edge, and we examined the expression of, and we did qPCR for quantitative expression of the cyclin E transcriptional factor. So we extracted the tissue, we extracted the RNA, we got cDNA, and we ran qPCR. Now, I have to warn you, these are preliminary results. It's been a little bit more challenging than what we expected to actually get high yield and high quality RNA, in part because of the fact that we are dealing with such little tissue. So in the coral animal, there are 2 tissue layers and then you hit stone. So it's a bit difficult. But right now, this is what our results look like. So of the 3 primers on the x-axis, we do see an increased, although not significant, but an increased expression of the cyclin E transcriptional factor on the edge versus the center. The interesting thing, so we're adding 3 more genotypes, and right now, literally as we speak, 2 of my students are in the lab probably doing an RNA extraction to add more genotypes to this data set. So the overall results so far, which is preliminary, is that we have increased expression of the cyclin E transcriptional factor on the edge versus the center. So again, we found edge tissue is the part of the microfragment that actually undergoes rapid growth, as well as we discovered that Orbicella faveolata does have the HIPPO growth pathway is present, and we were successful in developing the cyclin E primers, as well as increased expression at the edge, although there's only 2 genotypes involved. So, moving forward, what are we going to do? Well, we're hoping, of course, that there's some practical application. We do know that when they're doing these microfragments and they have all these different genotypes of all these different species, they do run them through some trials for increased pH, or decreased pH, increased temperature. Possibly, if there is differential expression between genotypes in growth, we might also want to target genotypes that have more rapid growth versus not as much. As well as increased number of genotypes, of course, we're going to increase that, and then target other transcriptional factors. We'd like to develop some other primers of other transcriptional factors that are involved in the HIPPO growth pathway and analyze those. As well as something that was sort of a surprise for me is that when I dissected the tissue, and then I came back to the same microfragment 2 weeks later, within 2 weeks, all that tissue regenerated, and I was blown away. And because we always think of coral being, and it is a delicate species, but at the same time, after dissecting that, I was shocked that this regrowth happened. So we've undergone we have some data that needs to be analyzed of the regrowth, but we also would like to go back and actually extract the tissue that had previously been extracted and look at the genetic expression in the injured tissue. So with that, I'd like to thank my team. So Dr. David Vaughn up in the right-hand corner, he retired from Moe, and he now has a nonprofit organization where he goes all over the world consulting. It's called Plants a Million Corals is the name of his organization. And then Sarah Hamlin at Mote Marine Laboratory, as well as my team here on the left. Molly Weglars and Natalie Villafranca are undergraduate students, as well as John Paul Dickenthaler. And then Hayden Tompkins was a postbac student in my lab. And then Dr. Drees Dennison, who is my collaborator at the University of Dallas. So thank you.